great to be here. Thank you for the invitation. I'm going to talk about a story that's been rumbling in my lab for a few years. Just getting ready to submit the paper. So I've been curious about mechanisms of drug craving for a number of years. What is drug craving? How does it work? There's various ways of trying to investigate drug craving, of course, in animals. But the approach I took, or the question I was most interested in, is actually the absence of craving. So you'll see in a minute what that means. But in the context of tobacco smoking, I think it's been known for many years that nicotine is considered a major component in tobacco smoke that drives addiction. And I love this paper actually. Every opportunity I get, I'll present it. This is a paper published by Finnegan in Science in 1945. And even back then, not almost 100 years ago, it was clear that smoking was problematic, that people who were smoking developed lung diseases, for example, even though the relationship between lung disease and smoking wasn't quite clear back then. When people tried to quit, they had difficulty. And so, again, that's been known for over 100 years. And what Finnegan did in the science paper was to try and kind of consolidate the mechanisms that may contribute to the difficulty that people have in trying to quit smoking. And some of the explanations, I think, are just fantastic and well worth ruminating over. So, why do people smoke? Why do they have difficulty quitting? Optical perception of cigarette smoke. They smoke because they like the smoke rings. Fire worship. So you smoke because it's your religion. Agreeable smell and taste. Anyone's ever smoked a cigarette and didn't continue to smoke? No, that's absolute rubbish. They taste and smell horrible. I'll leave the next one alone. The next one's my favorite. Allows one to do nothing gracefully. I think that's wonderful. You smoke because, it makes you look good. And of course, it's been known that nicotine is the major alkaloid contained in tobacco leaf for, again, well over 100 years. And the notion was that maybe the reason people smoke is to obtain some property of nicotine. Around about the same time, there were a number of very well-known kind of classic experiments where people would inject themselves with nicotine, including smokers. There was a number of physicians in London, for example, who carried out these experiments. And what they found is that the self-injections of nicotine were very reinforcing, to the point where they'd stop smoking cigarettes in lieu of self-injection of nicotine. So we've known for a very long time that nicotine is addictive. And so if nicotine is so addictive and so motivationally salient, why is it then animals don't really care if you take nicotine away? And so typically what will happen, there's two kind of key features of addictive drugs you'd expect to see in an animal if you make that drug available. The first is that the animal will volitionally consume the drug. It will be self-administered. And the second is, after a period of self-administration, if you take the drug away, the animal will be perturbed. It will have liked the drug, it will be motivated to obtain the drug, and when the drug is no longer available, the animal will seek the drug. And so nicotine is considered a relatively modest reinforcer because animals will self-administer it, but they consume at fairly low rates. Now, whether one equals the other, I think, is open to debate. Just because an animal responds at low rates does not mean the drug is not very addictive. It just means there's low rates of responding for whatever number of reasons. Heroin, for example, is consumed at relatively low rates because of the pharmacokinetic and the pharmacodynamic properties of the drug, but I don't think people would argue that heroin is not very addictive. Nicotine is the same. Because of the biophysical properties of the drug, it's consumed at low rates, but it's consumed nonetheless. But when you take the drug away, rather than an animal, and that animal could be a mouse, rat, or primate, what you almost invariably see is that the animals stop looking for the drug almost instantaneously when it's no longer available. And these are data from my lab. As an experiment, you'll see again these data in just a few minutes. We've seen this 100 times. This is well reported in literature. Animals with stable nicotine self-administration, you then switch the drug so it's no longer nicotine, it's saline, or whatever way you want to manipulate the animal. But whatever it is that you do, that nicotine is no longer available, the animal almost stops responding almost immediately. That's not what you see if heroin, if cocaine, if amphetamine is available. What you see is quite a marked increase in drug-seeking behavior. There's various names for this, an extinction burst, etc., etc. But the general idea is that the animal is motivated to obtain the drug that is no longer available, so will now invest effort in that hunting behavior. You do not see that with nicotine. And so the question we asked is, why? And that's important because the absence of that behavior has been used as an argument to suggest that nicotine is not an addictive drug, or at least is not the primary addictive drug or component in tobacco smoke, because if it were, animals would hunt the drug when it's not available, and they don't. And so we asked why. Anyhow, before I get to that, in this very same science paper, I remember reading it as a postdoc, something else struck me as being unexpected and kind of weird, and it resonated with me because I was like, what the hell is going on? And it is, when smokers consume cigarettes, what you see is an elevation of blood glucose levels. And you see that most readily when smokers are hungry. And I was struck by that. Why would nicotine have a metabolic effect, a glycemic effect? And it turns out what's going on is that when you're hungry, your autonomic nervous system gets turned on, of course. And the reason for that is you want to stimulate the release of glucose stores from your liver in order to maintain blood glucose homeostasis. That's a well-known counter-regulatory response. We know all the hormone steps, we know how it works. It's a stress response. And nicotine is able to enhance that response. So what you see in smokers when they smoke a cigarette and you get this glycemic response is basically a turning up of this sympathetically regulated glycemic response. And why I tell you all of that is because blood glucose levels are really, really easy to measure. And so we thought that this was a very simple, highly translationally relevant marker of the pharmacodynamic actions of nicotine. And this blood glucose elevation effect occurs in the brain. We published that a few years ago. So nicotine gets into the brain, it engages various circuits that are involved in stress. That will then send a signal to the pancreas, which will release glucagon, and that glucagon acts on the liver. And you get exactly the same response in humans that you see in rats, for example, that you inject with nicotine, or a mouse that you inject with nicotine. So we asked how much nicotine is required for an animal to self-administer during a self-administration session, to see the same glycemic response that we know occurs in humans. And the idea was, this is a very simple, straightforward measure to confirm that sufficient quantities of nicotine are being self-administered by our mice and rats to do what we know occurs in human smokers, which is an elevation of blood glucose levels. Again, very simple, but I think a very translationally relevant biomarker of target engagement and pharmacodynamic responses to the drug. So what did we see? Well, if you perform a self-administration, these are in rats, but you get the same thing in mice. If you perform the standard self-administration experiment that's been published, I would say probably thousands of times now, which is you let a rat intravenously self-administer nicotine at 0.03 milligrams per kilogram per infusion. That's the golden dose that we all use. You see no change in blood glucose levels after the self-administration, immediately after, relative to blood glucose levels assessed immediately before. And so why do we use that dose of nicotine? Well, there's no, it's not as if it engages the receptor in the brain, so you get a KPUU that's equivalent to what you'd see in a human smoker. There's no kind of basic kind of receptor autoradiography target engagement rationale for that. The reason is, back in the original self-administration paper published by Bill Corrigal's group in 1991, 0.03 mg per kg per infusion of nicotine was the dose that sustained maximal rates of responding in rats. And so by convention, essentially, that's the dose that we use in rats, mice, and monkeys. But that's the reason. And so actually, if you step back and look at physiological response to nicotine, using that dose that maintains maximal rates of responding, there's no change in blood glucose levels. If you increase the unit dose a little bit more to 0.09 mg per kg per infusion, once again, there's no difference. If you increase further to 0.12, 0.18, etc., much higher doses, you will get a very robust elevation in blood glucose levels, exactly like you see in human smokers. You will also find, if you look through the literature, that people who have mutations or allelic variants in various nicotinic receptor subunit genes, for example, the alpha-5 subunit, the alpha-3 subunit, the beta-4 subunit, if you recapitulate those mutations in mice or in rats, you will not see a nicotine-related phenotype at the lower doses that do not elevate blood glucose levels. You only see altered nicotine self-administration in rodents that bear these mutations that are relevant to smoking behavior when you use these much higher doses of nicotine. So that's established in literature. It's been known for about 10, 15 years now. So the reason I'm showing you this is I think this may suggest that we've been using the wrong doses of nicotine. And if you elevate the doses just a little bit more, you now begin to see that the genetics that we know influence tobacco smokers and smoking in humans now influences tobacco use in rats and mice. You now get a physiological response that's been known for over 100 years in smokers will now occur in rats and mice. So again, I show you all of that because once we settled on doses that we thought were now relevant to what's happening in human smokers, we then asked, well, what happens to behavior? And of course, the very first behavior we looked at was craving-like responding. And so we did that experiment, which is we allowed animals to self-administer nicotine, either the dose that we know and love, and the entire field uses, or a slightly higher dose that recapitulates this glycemic response to nicotine in a rat that we know occurs in human smokers. And what we saw was basically diametrically opposed effects. If you look at responding in animals that have the standard dose of nicotine, baseline is when the drug is available, and then the days of withdrawal are essentially you've switched nicotine with saline. It doesn't matter if those of you who are self-administration aficionados, you have your Q light that comes on when the animal infuses nicotine, etc. It doesn't matter whether the Q light is turned on or not. If you take nicotine away, responding extinguishes, and then with the higher dose, if you take nicotine away, what you see is a very robust increase in drug-seeking behavior that typically lasts anywhere from 2 to 7 days, sometimes even longer. In other words, completely the opposite effect that we see with the standard dose. And now nicotine is beginning to behave itself. It's beginning to do what you'd expect to see from a drug that's actually contributing to the motivational properties of cigarettes. Okay, so if that's true and these higher doses are actually relevant, that means that perhaps higher doses than we typically use when we're trying to understand what nicotine does in the brain may be worth looking at. And so that's what we looked at next. In other words, what do hyperglycemic doses of nicotine actually do in the brain relative to the types of doses that we and others have been using for decades now? So one of the first things we did was simply inject mice. So this is an acute injection of nicotine, either saline, a standard dose of nicotine that doesn't modify blood glucose levels, or a higher dose that will trigger the same hyperglycemic response. So these are not self-administering animals, this is not chronic drug exposure, this is just a higher dose of nicotine that we think begins to recapitulate the actions of nicotine that occur in the brains of human smokers. We killed the animals 2 hours after the injection, and we did whole brain CFOS staining. So simple CFOS mapping to see if anything looked different with this higher hyperglycemic dose of nicotine that we think is more relevant to what occurs in the brains of smokers. And so what I was looking for personally was, okay, maybe you'll get some brain regions where perhaps we'll get a nicotine-induced FOS response with the standard dose, but surely there'd be brain regions where you're going to get more robust response with the higher hyperglycemic dose, and those regions that come online more vigorously, if you will, with the higher dose of nicotine, maybe those that are contributing to this craving-like behavior during abstinence. So that was the aim of the experiment. And what we found were kind of 3 different patterns of FOS responses. All the areas basically stacked on top are those where there was no kind of dose dependency. So in other words, relative to saline-injected animals, a nicotine injection, regardless of the dose, would increase CFOS. And there really was no difference in terms of the standard or higher dose, the hyperglycemic dose of nicotine. And there's a whole bunch of areas that come up all the time, particularly cortical regions, that looked pretty much the same. Then we got the regions that I was most interested in, which are those that seem to demonstrate this dose dependency, where a higher dose of nicotine induced more CFOS. And the area on the top of the list there, the interpeduncular nucleus, is an area that my lab works on. It's got some of the highest densities of nicotinic receptors in the brain, particularly those nicotinic receptor subunits that are known to be associated with risk of tobacco smoking. So these were the areas that at least initially we were most interested in. Then there's another area there called the triangular septal nucleus, and that was kind of an anomaly. There was only a very small number of regions where we got something kind of weird, which is we would see a FOS response mounted in response to the lower dose of nicotine that seemed to go away when we gave the animals a higher dose of nicotine. And of course, we're looking at, this is unbiased, you map your brain regions to the Allen Brain Atlas, and you're scanning throughout the entire brain, so you're quantifying CFOS expression, 230, 240 discrete brain regions. You got like 5 or 6 animals per group, so of course there's plenty of occasion for errors to creep into your experiment. It's not properly powered to do that. You'd need to probably have like 50, 60, 70 animals per group, which we've not done. So initially, when I saw that nicotine at the higher dose was simply not doing anything in this region and a couple of others, I was like, what would you expect when you're looking at over 200 brain regions? Of course, you'll have some kind of error, some kind of weirdness. But I'll get onto that in a second. I just wanted to, I'd never heard of the triangular septal nucleus, had no idea whereabouts it was, besides probably somewhere in the septum. But it's actually located in the posterior region of the septum. So when I think of the septum, I think of the medial septum and those cholinergic neurons that project into the hippocampus, classic septohippocampal system involved in memory and learning. And then, of course, you got the return pathway, the reciprocal pathway, the reverse arm, which is from the hippocampus down to the lateral septum. And the lateral septum are GABAergic, so classic septohippocampal loops. The triangular septal nucleus is not part of that. It's in the posterior septum, and we know next to nothing about the posterior septum. So I didn't feel too bad about not hearing of the triangular septal nucleus before, because there's probably only maybe 2 or 3 brain regions or papers ever published about this region. Anyhow, just to show you, this is kind of like a heat map, if you will, of the phos plume that we observed. And so relative to saline animals, hopefully you can see that there's more yellow and orange, in other words, more phos in the triangular septal nucleus, in response to the standard dose of nicotine, whereas the higher hyperglycemic dose appears to, if anything, desensitize this region. Anyhow, I tell you all of that because we did the more complicated experiment next, which is that's mice acutely injected with nicotine. We then prepared groups of animals that self-administered either saline, the standard dose of nicotine, or this higher hyperglycemic dose that I'm trying to argue to you may, to some degree at least, better recapitulate what nicotine is doing in the brains of human smokers. We took animals after the self-administration session, we anesthetized them, then we used functional magnetic resonance imaging to try and identify regions that showed differential activity, at least in terms of Bol signal. And we did that, and what we saw, I think you see here in red, red means more neural activity, more Bol signal when animals had the higher dose of nicotine relative to the standard dose. Exactly what we thought would happen. We see lots of changes, lots of increased activity in somatosensory cortex, for example. You might expect that considering these animals are taking a big bolus of nicotine relative to the standard dose. So that would have some somatosensory properties. But we found a whole bunch of regions where those areas are coded in blue. They are areas where you get lower neural activity when animals take these hyperglycemic doses of nicotine that trigger craving during abstinence relative to the standard dose. And I did not expect to see that. Having worked on nicotine in grad school, I just always think of nicotine as being incredibly excitatory. It just turns on the brain. Okay, you can desensitize nicotinic receptors, but generally the response is almost always excitatory and persistently excitatory. Here, when we do the imaging, we see this decrease when animals get more nicotine in terms of neural activity. And it turns out one of the areas that was smack right in the middle of a node of decreased activity in response to these hyperglycemic doses was again the triangular septal nucleus. So here are 2 different species, 2 different modes of nicotine delivery, 2 different ways of measuring brain activity, and the same region came up twice. And so that, of course, is of interest. What I'm not going to show you is we did in vivo ephys, photometry, etc. And this region is very, very sensitive to these higher hyperglycemic doses of nicotine. So we decided to focus on this part of the brain to see if it may contribute to this craving-like behavior we're seeing. And this just quantifies what I just said, which is the standard dose of nicotine actually will increase neural activity in this part of the brain, the triangular septal nucleus, relative to saline self-administering animals. And with the higher dose that will elicit craving during abstinence, we get the opposite effect. Okay, what is this brain region? Like I said, we know next to nothing about it, so we decided to see if we could map it using spatial transcriptomics to understand which neurons are contained in this brain region, what's their phenotype, which kind of flavors are in there. And so if you look in the Allen Brain Atlas, what you find is that a nice marker gene for this brain region is calbindin-2, calb2. So we used xenium. This is xenium 1.0, and that has limitations I'll talk about in a minute. And major limitations, we can only map about 300 to 400 genes using this procedure. Anyhow, one of the genes that we confirmed we could detect is calb2. I hope you can see that. These kind of green cells are those that express calb2, and that kind of mapped on to anatomically where we expected to see the triangular septal nucleus. So we're fairly confident that we've captured that area in our xenium experiments. And so we then looked at the calb2 positive cells to see what they'd express. And then we did your typical U mapping, which is not just those cells, but all the cells that we captured in this brain region. How do they stack in UMAP space, based on the genes that they express? And we found a whole bunch of glutamatergic neurons, the bottom that express calb2. And they were exclusively glutamatergic. All that to say that the triangular septal nucleus neurons are not cholinergic like the medial septum, they're not GABAergic like the lateral septum, they're excitatory, they're glutamatergic. And in fact, if you look, you can see that there's kind of blue and gray flavors of these cells. And the reason for that is there's actually two subpopulations. Tiny little brain region, but already in there, there's two subpopulations. They all basically express VGLUT2, but about half the cells, perhaps maybe a little less than half, also co-express VGLUT1. And so two populations, they're both glutamatergic neurons. We did these experiments in control animals and animals that were self-administering higher hyperglycemic dose of nicotine, that we confirmed were craving the drug, and we saw the spike in drug craving. That's when we killed them. And then we compared gene expression profiles between the saline animals and the nicotine craving animals. What we found is that even though it's a limited gene kind of probe set that we looked at, I can't remember, 350 genes, there was all the genes that are involved basically in glutamatergic transmission or intrinsic neural activity were dysregulated in the nicotine animals. So nicotine is doing an awful lot in terms of transcriptional remodeling of these cells. And some of the genes that were impacted are those that are involved, oh, I better move quickly, in glutamatergic transmission. Anyhow, if that's important, if this region is important, if it gets, turned off by nicotine, if we turn it back on again, we'd expect to see some impact in craving. So we chemogenetically stimulate this region. It turns out when we did that, we killed most of the animals. And why did they die? Well, they drank themselves to death. It took us a while to figure that out, believe it or not. It turns out the reason being that the subfornical organ, the major thirst organ in the brain, is right beside the triangular septal nucleus. And death is a major confound in behavioral pharmacology experiments, so that didn't work for us. So then I've got 5 minutes left, I'm going to move quickly. We tried to take a circuit-based perspective. We can't directly manipulate these neurons because we're directly impacting the subfornical organ. Let's take a circuit-based mechanism and manipulate the circuit that these cells are engaged in rather than the cells directly, so we can avoid impacting the subfornical organ. So we used a procedure called MapSeq, which, again, not to belabor this, we can basically barcode with individual genetic barcodes, triangular septal nucleus neurons, and then we can find where in the brain those neurons are expressed. So you get single cell neuronal mapping. We did all of that. We basically looked through all brain regions, either candidates or those that we suspected from literature are direct targets for triangular septal nucleus neurons. And all of that basically distilled down into two major pathways. Triangular septal nucleus neurons either go down into the basal forebrain and into the hypothalamus, or they go up into the habenula. And for me, the habenula was a great find because we already know that the habenula plays a key role in regulating the motivational properties of nicotine. If we let animals self-administer a hyperglycemic dose of nicotine, what happens is the triangular nucleus neurons that are projected at the habenula basically get turned off. So we thought this circuit is likely important. Nicotine seems to inactivate not just the triangular septal nucleus, but inactivates the projections in that region to the medial habenula. Is that important? Yes. If we lesion or chemogenetically inactivate this circuit, we get more craving. If we chemogenetically stimulate, we block craving. So it turns out that through dumb luck, we stumbled on a brain region and a circuit that seems to play a key role in regulating nicotine craving during abstinence. And there's a strange link between glycemic regulation and this craving behavior, which I'm not going to go into, but keep that in mind for just 1 second, and I will finish in just a minute. So I said one of the limitations of Xenium that we used, at least, we've now gone back and used some other kind of more recent spatial platforms. The platform that we used had about 350 genes, very limited, so we decided to map this region using single nuclei sequencing. We get like higher resolution gene expression profiles, which is what we did. There's calbindin2. You can see a nice cluster of calb2 expressing neurons. They also express other gene markers of triangular septal nucleus neurons. I show you all of that because what we found, again, through dumb luck, is that these cells also happen to co-express a whole ton of GLP-1 receptor. And we looked for GLP-1, not thinking it was in there. We looked for GLP-1 because I remembered that it was heavily enriched in the lateral septum, lateral septal nucleus, which is right next door. So I wanted to confirm that we had lateral septal nucleus in one cluster, which we do. You can see it right there in the middle. It turns out there was a lot of GLP-1 receptor in the triangular septal nucleus. For me, that was really intriguing because my lab's already published a bunch of papers suggesting that GLP-1 signaling in the habenula and IPN regulate nicotine craving, regulate nicotine seeking. That's problematic for me because it turns out we went back and spatially sequenced and single nuclei sequenced these brain regions. There's no GLP-1 receptor in these regions. even though we published a bunch of papers suggesting that GLP-1 signaling here is important. It turns out the triangular septal nucleus, which projects these regions, does have GLP-1 receptor. So we think that we can explain these previously published findings by virtue of the fact that the GLP-1 receptor is probably presynaptic on these septal inputs to these regions. Okay, is it really there? Just the RNA scope, calbindin 2. I hope that you can see red. There is a lot of red GLP-1 receptor. There is a ton of GLP-1 receptor in this brain region, which has not previously been reported before. That's intriguing to us because this region seems to regulate craving. And I think we're all aware now of the Ozempic story, anti-craving effects. We don't know how they work. So of course, like, maybe we've been doubly lucky and we've stumbled upon not just a craving region, but a region that may regulate the anti-craving effects of Ozempic. We confirmed that GLP-1 receptor is also in this part of the brain in humans, so it looks like it's translationally relevant. If you remember, I said there was 2 flavors of these GLP-1 neurons. If you use RNA scope, it turns out only the VGLU2 cells in this region, the subpopulation that expresses VGLU2, they express GLP-1 receptor, not the VGLU1 flavor of cells. And it turns out that if you stimulate these VGLU2, or if you record these VGLU2 cells, they've got different firing modes. Ozempic very much turns them on. So these receptors are in this part of the brain are functional. These GLP-1 expressing triangular septal nucleus neurons, where do they project to? We use the GLP-1 Cree animals to tag them fluorescently. They project exclusively to the habenula. And if you stimulate these cells and then expose them to Ozempic, what happens? You boost excitatory transmission in this circuit. This is the circuit that gets turned off by nicotine during craving. So we asked what would happen then, of course, if we were to turn this circuit back on using Ozempic, seeing as Ozempic is able to do that. So we simply injected Ozempic into the triangular septal nucleus. We took nicotine away, hyperglycemic dose of nicotine in control animals. Craving effect is there. It is completely gone. And we stimulate GLP-1 receptors in this part of the brain. And there is no effect, as far as we can tell, on appetite when we stimulate these receptors. So we do not know what GLP-1 is doing in this part of the brain. We suspect it's probably part of some feedback mechanism that's involved in glycemic regulation, something we're testing right now. I'm finished. I just want to thank everyone in the lab. Thank you very much.